Hi, and welcome back. In this video, I'm going to talk about cell division that includes mitosis and meiosis. Before we get into the details, let's understand what is a cell cycle. A cell cycle refers to the normal activity pattern of a cell. As you can see here, it is divided into two phases. First is the interphase, and next is the mitotic phase. Um, the actual cell division, which is mitosis and cytokinesis, is occupied by a very small portion, a very small pizza slice out of the entire process that the cell undergoes to divide. Interphase includes different other phases or stages like G1, S, and G2. G1 and G2 includes the growth phase, and S phase is the synthesis phase. S stands for synthesis, and that is where the cells replicate or the DNA is replicated. Mitotic phase is um, the phase in which the cell actually divides. Mitosis includes the preparation for cell division, and cytokinesis is when cytoplasm divides. Cyto means cytoplasm or the cell, and kinesis is division. So the actual cell divides. There is a difference between animal cell and plant cell division. In animal cell, if this is a cell, it forms a cleavage furrow from the outside to inside this part, and then it separates into two different cells. And in plant cell, the Golgi vesicles accumulate in the center of the cell to form a cell plate. As you can see here, it forms a cell plate which widens outward to separate out the two different cells. Now let's move forward to the cell division. It is a process by which all living organisms grow and reproduce. It is important for growth, development, and repair, and also for reproduction. There are two types of cell division, including mitosis and meiosis. Mitosis is for somatic cells like blood cells, nerve cells, muscles, and skin cells. And meiosis is for reproductive cells, which includes sperm cells and egg cells. This is what cell division typically means. A single cell divides into two, and those two cells individually divide into two each, and cycle goes on. Now let's understand the important terms necessary for this particular lab. First is chromosome. It is a thread-like structure carrying the genetic material in the form of DNA. This is a structure of a chromosome. This entire structure is known as chromosome. Chromatid, it is one of the two strands of chromosome. So this blue colored part here is one chromatid, and pink color or red colored part is second chromatid. So two chromatids combine together to form a chromosome. Centromere, it is the constricted region of chromosome which helps in cell division. So this yellow colored attached space here is the centromere. Next is haploid. It is the single set of chromosomes, often referred to as N. And diploid, it is the two sets. So two sets of chromosomes combinedly are known as diploid. It is referred to as 2N, which means one from each parent. Crossing over, it is an exchange of genetic information between chromosomes, resulting in non-identical chromatids. So this is how it takes place. Homologous chromosomes, which means two identical chromosomes, one from mother and one from father, um, cross over. This is the actual action of or the step of crossing over. One of the strands or one of the chromatids exchange their genetic information to form a chiasma. The X-shaped structure that is formed over here is known as chiasma to form recombinant chromatids, and that's how the crossing over process is taking place. Let's get into the overview of mitosis. Definition: a type of cell division in which a single cell divides to produce two genetically identical daughter cells. The parent cell undergoes DNA replication so that the single chromatids that you see here are forming the second replicate of themselves and then dividing into two daughter cells. We'll understand how this happens in the further slides, but this is just an overall picture of mitosis. Purpose: it is for growth and repair of tissues and replacement of old or damaged cells. So when it comes to wound healing, this is what occurs in the skin cells. Now let's understand the process of mitosis. First step is prophase, which means the very initial step. It is often separated into early prophase and late prophase, but for now it's okay if you just understand it as a complete process of just prophase. In prophase, chromosomes condense, spindle fibers form, and nuclear membrane breaks down. So these two structures are the centrioles, and this is the spindle fiber. Uh, the dotted line over here is the nuclear membrane. The blue chromosomes are from one of the parent, and green ones are from the other parent. The black dots in the middle are centromeres. Okay, so metaphase. Chromosomes align at the equator. If you can see here, all the four chromosomes have aligned themselves at the equator, and because there is no nuclear membrane now, they are out in the cell, and the spindle fibers have attached themselves to the centromere from centrioles. Anaphase is when sister chromatids are pulled apart to opposite poles. So anaphase. So here, the two long ends of the chromatids are known as sister chromatids, and the two short ends are known as sister chromatids. But this and this is not sister chromatids. And the green long end of the green chromosome and long end of the blue chromosome would be non-sister chromatid, because both of them were derived from different individual, like one from mother, one from father. But if it is the same chromosome, it's going to be sister chromatids. And if it's different, it's non-sister chromatids. So in anaphase, the sister chromatids are separating. See? Yeah. In telophase, chromosomes reach the poles, nuclear membranes reform. As you can see here, the chromosomes are at the opposite poles, so that there are no longer any spindle fibers attached anywhere, and the nuclear membranes have reformed, the small circles that you see here. Next step is cytokinesis or cytokinesis. It is the step in which cytokinesis is the step in which cytoplasm divides. Cyto is cytoplasm or cell, and kinesis is division, forming two identical daughter cells. So this constrict grows and separates the two cells or two daughter cells. Moving on to the overview of meiosis. Definition: a special type of cell division that reduces the chromosome number by half, producing four genetically diverse haploid cells or gametes, because meiosis typically takes place in reproductive cells like gametes or sperm and egg. Purpose is formation of gametes, as I just mentioned, introduce genetic variation through recombination or crossing over. Cell replicates its DNA, which divides into two through meiosis 1. There are two parts in meiosis 1 and meiosis 2. So this is the first stage, meiosis 1, and then those two individual daughter cells divide themselves into two other daughter cells. So in total, from one parent cell, there are four daughter cells forming, which are not genetically identical. They are genetically diverse. So next is the process of meiosis. Homologous chromosomes pair up and exchange the genetic material in prophase 1. As you can see here, synapses and crossing over is taking place. As I just explained in the introduction or the important terms slide, what crossing over is, it is the exchange of genetic material between the parents. Metaphase 1 is when the homologous pairs align themselves at the equator. Same thing happening, they are aligned at the equator. Anaphase 1, non-sister chromatids are pulled apart to opposite poles. In mitosis, it was the sister chromatids, and in meiosis, it is non-sister chromatids. It is the opposite colors that are being pulled together or pulled apart. Telophase is when the cells divide into two, each with half the number of chromosomes. And remember, they are still duplicated. As you can see here, it is still duplicates. Meiosis 2. Meiosis 2 is pretty much similar to mitosis, but it results into 4 haploid cells, each with one copy of each chromosome. So if you see here, yeah, everything is happening just as mitosis. Only difference is that there are 4 haploid cells forming because 2 cells are undergoing mitosis at the same time and not just one cell. And remember this, that meiosis 1 begins with a diploid cell, which is 2n, and it leads to haploid, which is it leads to 4 haploid cells with single set of chromosomes 1n. Here are the key differences between mitosis and meiosis. First is the cell type. Mitosis occurs in somatic cells or body cells, and meiosis occurs in germ cells or reproductive cells, which is gametes. A number of divisions is just 1 in mitosis, resulting in 2 cells, and number of divisions in meiosis is 2, 1 and 2, resulting in 4 cells. The genetic composition of daughter cells in mitosis is identical to parent, while in meiosis it is genetically unique or genetically diverse. It is not the same as the parent cell. Chromosome number is same as the parent cell, which is 2n in mitosis, but as we just saw, in meiosis it is different. It is reduced by half, leading to n number of chromosomes or haploid chromosomes. Function is for in growth, repair, and asexual reproduction for mitosis, and sexual reproduction and genetic diversity in meiosis. Thank you. Hope it was helpful. And as a reminder, my office hours are Mondays 1:30 p.m. to 2 p.m. Thank you. Bye-bye.